Hello, this is James Tsai. Today, I'm going to talk about bioinformatics and its applications in pandemics and infectious disease. I'm going to mention two different pathogens. The first one is called Penicillium magnificae. It's a fungal species. The second one is the SARS-CoV called coronavirus 2. So for the first one, I will mention how the genome sequence has been determined for this unknown species, and how are you going to analyze the genome sequence using computational approach. And then I will mention how can you design an RNA sequencing experiment to tackle some of the transcriptome information from the species. For the second part, I'm going to mention machine learning analysis using single cell data and to establish gene regulatory networks, and then using the method to study the potential mechanism of action of anti-malaria drugs against SARS coronavirus 2. Okay, so for the pathogen genome sequencing here, I'm going to tell you about the sequencing technology and the genome assembly. And the first is our species. It's called the Penicillium magnificae. It's a fungal species discovered in 1950s. And it's so-called thermodimorphic fungus. I will tell you what it is. So under different room temperature, this kind of fungus can grow in different forms. At the room temperature, which is 25 degrees C, this is a mold form, and it's filamentous fungus, and so you see the mycelium cells. But when the temperature increases to 37 degrees C, which is the body temperature, and this particular fungus will grow as a yeast, this unicellular form of the growth. And this is an optimistic infectious pathogen, and it's particularly dangerous for AIDS patients because they have a compromised immune system, and it gives the chance for the Penicillium magnificae to get into the blood and cause the systemic infection. It's caused pandemics in Southeast Asian area, and it's nature reservoir in bamboo rat, and that's the Southeast Asian. So that's what the colony look like in the petri dish, and that's 37 degrees C, and this fungus species grows on the algae, and you see this typical yeast colony form. But when you move this particular colony to room temperature, 25 degrees C, and you will realize after 1 week it grows into a mold colony, a mold, and also has this red pigment around this colony. So very characteristic features of this. And this is a experiment we did in the lab. Transit a plate from a 37 degrees C into room temperature, 25 degrees C, after 2 hours, 3 hours, until 24 hours and 1 week. We have 3 replicates, and you can see the change of this colony from one growth form to another, from yeast transit to mold. And this is under microscope, and you see mycelium cell and yeast cells for the same species. Is very special, and this is called thermodimorphism. Why we are interested in this growth form change? Because in the systemic infection caused by penicillin manifae, it has to be yeast cells, and it grows inside human cells. Okay, so if we can somehow control the transition from mold form to the yeast, so if you have a interruption, can control the growth form and stop the change, then we probably can control the infection of this particular fungal pathogen. So we would like to understand whether there's a temperature regulated gene, and also maybe there's a sensor. It could be RNA-based thermal sensor to allow this mycelium cell to sense, to feel the change of the temperature and then to switch the growth form. We would like to understand the molecular mechanism behind this. It could be the secondary structure change. So that's the background, and the approach we are going to use is genomics based. So we want to have this fungal has been sampled, but when we started the study, we have no genomics information. We don't know the composition of their DNA. So what you can do, and that's also the typical preparation of genome sample. So you can purify genomic DNA out of it, species. And then there are steps of a preparation. At the end, you want to prepare so-called genomic DNA library. So this library, as a sample you want to put into the sequencer, so you get the fragment of DNA, then you put them back. So you reconstruct or assemble a genome. So that's the typical procedures, the workflow. You prepare your genomic sample in order to get the genomic DNA library for sequencing. I will quickly mention the sequencing technology. So there are 3 generations of sequencing technology. First one is based on the Sanger sequencing, then you run the GL at very early age. And then there are machines in to do this, and so-called Sanger sequencing, and the Sanger has won the Nobel Prize twice. The second time is he created this method, invented the method to sequence DNA. And then we have so-called secondary, second generation of sequencing technology. We also call it next generation sequencing. And after many years of development, now Illumina is the company dominant the whole market. So when we say we are doing next generation sequencing, most of the time we are going to load our DNA RNA sample into this machine produced by Illumina. And the Illumina machine will generate this large amount of this DNA samples sequencing data for you. And the next generation sequencing technology, also called third generation. Now the representative companies are this so-called Oxford Nanopore and also PacBio, these 2 major companies. I don't know about these 2 companies right now, but these are the 2 most important companies. Technology is for the third generation sequencing. So what good about the third generation sequencing technology and is it can generate very long reads. The reads is a fragmented DNA you can generate. And if you can generate very long reads, in this case, Oxford Nanopore technology can generate on average over 1,000 or maybe 1,500 base pair, and even 10,000 longer rates. It will be very useful when you have unknown species. You want to know the whole structure of this genome sequence. Then the long rates will be really helpful for you to understand this genome structure. So this is a machine for Oxford Nanopore, and you connect it to a computer, and you load your sample into DNA sample into this so-called cells, and then put the cells into this cartridge. Then you can read out and use a technology, the nanopore technology, to read out the DNA directly. Anyway, we with all this sequencing technology, we will generate a large amount of sequence, and they are in the files. And next you want to do is to either, if you have a reference genome, which you know the genome of the species, you can map these rates to the genome and to identify the variations, the difference between the sample you sequenced with reference. Or if you don't have any reference genome, you want to reassemble the genome from scratch. So after that, you have a additional data analysis on that. So this is a concept map here. So this is a technology in the second generation sequence. It's a synthesize based, and also it generate this so-called clusters on the sequencing machine, and they take pictures and then convert it into sequence. So this is laser-based pictures. Anyway, so you won't be able to, you don't need information of this original image generated by the sequencer. You don't need the intensity files. All you will get from the sequence machine and useful information are these rates, and then your downstream analysis, including those kind of alignment, you want to put them together and assemble them, or you want to compare and see with reference. So that's what the informatics, bioinformatics play a role in this analysis. And then you have data management problem, the QC, or the downstream analysis. Now, once you have those short rates, because next generation sequence just cannot get the long enough DNA fragments, it will generate a lot of those short elements, short fragments. The first thing you want to do is to reconstruct, it's called Tandi Assembly, to reconstruct its original DNA sequence. It's along from the species itself. So this is an algorithm has been largely solved. The Tandi Assembly, this example, you have a short rates, and then you found the consensus, and then put them together. This is the consensus contig. So you like rebuild the puzzle, and then you put them pieced together and to get the original sequence, where this is from the genome samples you get. There are a lot of software tools has been built, and the CAP3, FRAP are used in the early stage, and there are new generation assemblers, and can be applied to large amount of the data directly. So the basic steps, you overlap the short rates, and then lay out into contigs, and then build the consensus and get the consensus sequence. So at the early stage, there are clone-based shotgun sequence, and then the whole genome shotgun using second generation technology. If you do the genome sequencing, sequence unknown species is called de novo sequencing or assembly. The strategy we are going to use is called hybrid strategy. That's a combination of second and the third generations of the sequencing technology. Remember, second generation sequence are short reads, but you can generate a larger amount using a relatively affordable resource. Third generation sequence is more expensive, but you can get much longer per unit reads. So that will help you to lay out this scaffold and to reconstruct the whole genome. So the hybrid strategy is the most advanced technology today used in genome sequence and assembly. So this shows you the strategy, the hybrid assembly. We have short reads and long reads provide, and then we can correct the errors in the long reads and then assemble the long reads, because long reads generated by for example, Oxford Nanopore, the third generation sequencer contains a lot of errors, and you can align the second generation sequence short reads to longer reads to correct those errors. Then you assemble these longer fragments into genome. Okay, suppose you have the genome has been sequenced and assembled. So now you have the reference genome. Then what you will have to deal with bioinformatics problem is the gene annotation. You want to know what gene has been encoded in the genome. And then you want to do some comparative genomics analysis to compare your species against other species, or to reconstruct the evolutionary relationship, genetic distance between species or between genes, and to understand your species and transfer some of knowledge in other model organisms apply to your unknown species. So for example, the penicillin manifold, once we get the genome assembled, we are going to do this so-called gene prediction and annotation. Here, we emphasize the protein coding genes. The problem has been largely solved because the protein coding genes have a specific sequence motifs. You know, a lot of this translational starting site is ATG, and they are coding stop codons. So GeneScan is the software tool that can identify the complete gene structures by using the genome DNA sequence alone. And then, for example, you can put your fragment, the genome sequence in this tool, their website interface, and you will get annotation to tell you where the gene is and what's the gene structure, intron and exon are. And then you can use a tool called Artemis genome browser and annotation tool to annotate manually to change some of the structure. So this is a screenshot for GeneScan web server. So you put your sequence in this and click a button and select what species you want to predict the gene structure. And then this web server will generate the output of where the gene exons, introns are, so the structure of the genes. Then you can visualize, use Artemis, a tool like this, Java-based. And then this is a positive strand, the negative strand, and all the genes has been annotated based on the automatic annotation tool, the prediction tools. So this is the first round, computationally predict gene annotation. Then you can do the manual adjustment to make it more accurate based on your other experimental information. And then the next will be the comparative genomics. We want to compare your gene with other species. For example, this particular gene protein coding sequence against other species. So it is highly conserved. So we see there are strong levels of similarity between the same gene across different species. And then you can study the structure, the order, orientation, and the neighbor genes and how they arranged in the genome and compare across species. That's a penicillin manipulae compared with Aspergillus fumigators, and you see the similarity between their one of the gene sets. And then in fungal species, a lot of genes under the same biosynthesized gene clusters, the same group of genes will be next to each other. So this from the gene island and the clusters. So you compare across different species and you know the function of the gene. Once you know one of the genes function in one species, we translate the function to this unknown species, cross species translation, transfer of the knowledge. So that's the comparative genomics. In our case, we understand this particular genes knockout and study its and to compare the prediction from the gene sequence annotation and the conformance function. And after knockout of this gene and the pathogenicity of the species of penicillin manipulae decreased. We can predict protein function, the gene product function, using a series of tools to study, say that, for example, the signal peptides and the transmembrane region prediction and the post-translational modification. There are different algorithms based on the pattern recognition and the weight matrix, sliding window, etc., can help you to perform this performance. So this shows you the sliding window analysis. That's your predicted protein sequence of the gene. Then you can, using a sliding window, then compute the score. Then you move to another space window and then compute some of these scores. In this case, is a hydrophobic score, and you can tell which region and there is more hydrophobic in the protein sequence. And then this analysis, for example, if you can predict the surface exposed portion of the protein, you may use that information to help to develop a vaccine. Okay, there are also efforts of the so-called genome resequencing. In that case, you want to identify the difference, base pair difference between subspecies. For example, if you know the genome sequence of the reference, and then you want to know particular strain and whether there are new mutation in the strain. And you can also use that RNA sequence analysis to do the transcriptome analysis. So there are some high level of application area and for next generation sequence. And you can work on the genome, you can work on the transcriptome to do, you can also work on the epigenomics level and also metagenome level, and that's microbiology. There are a lot of software tools you have to be familiar with before you know how are you going to combine them to achieve some of the goal. And there are aligners to do the alignment. There are gene expression analysis tools and expression study. And then if you're interested in the alternative splicing, there are particular tools that can be used. And I will skip some of these details. That's the fast files and its quality files to give you the base pair information. And then each quality score tell you the accuracy level, and you want a higher score because you want to be highly significantly accurate. And then they're FASTQ format file and similar to FASTA, but the quality score is encoded by this characters, and the sequence itself is here. And that's the overall map of the resequencing analysis. And then you have the raw data and the short reads, and you want to do the so-called read mapping, and then the downstream will be based on the mapping results. So once you mapped the reads to reference, that's the file sum and the BIM file you will use to keep or store this sequence alignment. That's a structure you don't even have to see, because there are tools, so it's called sub tools, and computational program to help you to manipulate those sum and BIM files, their alignments. So this is applications. For example, you want to view a BIM file stored in remote server, FTP, and you want to look at the particular region, that's chromosome 6 from a base pair this to here. Then you want to see what's the structure look like, and then they give you something like that. You can also have highly interactive genome view to visualize these BIM file alignments. And then in this case, you'll see a normal and a tumor sample side by side, and the coverage of the map significantly decreased in the tumor sample that indicated in the genome there's a deletion of the fragment. You can use that to detect the SNPs, so-called single nucleotide polymorphism. You will identify the difference between individuals or different chromosomes. So you will store the genetic variation information in the so-called VCF file. It's a variation file. It tells you where the variation is chromosome and position, and also the reference and alternative. In this particular SNP, you have AG change. You can count how many mRNA reads have been mapped to a particular gene. You can measure gene expression level, use so-called RPKM. And this is one example to show you how the RPKM is computed. And then once you have this, so after the mapping, that's options of different tools you can do the mapping. After the mapping, you have to summarize the data to compare between species, for example, and then between different samples. And then you have a differential expression analysis. There's different tools you can use. And the pipeline options for the RNA-seq analysis. So this is for RNA sequencing. RNA sequencing is transcriptome sequencing to compare gene expression difference across samples, between samples. You can use so-called top hat, cuff links, cuff diff. There's a series of the packages. And you can use bow tie, summate, or another tools to do the DE analysis. So there are different combinations, different flavors can achieve similar goals, and there's options of pipeline of software. And of course, you want to do some QC here. And that's top hat cuff link software package, and where you can download them. This is a little bit old, but still some people still using it. And that's a summate interface, and you can use this to do a lot of the things and help you, because there's an interface and it's Java based. Okay, in our case, we want to study mRNA, the gene expression change, and of penicillin malinifer at a different temperature. So we have samples from east cells grows in 37 degrees C. We also have samples from mycelium cell, which grows in 25 degrees C. Also, we have a transition phase of the cells. So we have 4 samples. And then we start with the 2 samples to compare cells growth in 25 degrees C against 37 degrees C, see whether there are change in the gene expression level. For example, this gene ABAA is downregulated in high temperature. And that's some of our discovery. We put them together and see whether there's consistent pattern and a set of genes up or down regulated. Now we can identify the differentiated genes and differentially expressed genes using the pipeline I just mentioned. And confirmed with some previous known information about the gene function, which phase this gene most likely to be involved in. I also studied the gene expression of gene clusters. They have very consistent expression profiles. So all the genes in this cluster expressed similarly, regulated in the same direction. And then we can work on the gene set and see whether upregulated gene and the downregulated gene play different roles. And this is called the gene ontology analysis. So when you have one gene, you may not be able to recognize the biological process involved, but once you have a lot of genes, you put them together, they regulated under the same rule, then you have a synthesized information about the function of genes as a group. And then because we have 4 samples, then we can see some samples will have a low expression at 25 degrees C, high expression, and change very quickly, or other patterns you can rebuild the reclassified all the genes into different clusters, different groups, and then look at their significant gene set analysis to understand this function. Okay, so for the rest of the 15 minutes, I will switch to another topic, which is we are going to use machine learning algorithm to study single cell data to build gene regulatory network and apply to COVID-19 analysis. Now we are at an intersection of 2 major technology revolutions. One is machine learning, another is called single cell technology. Traditionally, as we mentioned in RNA-seq, per sample, you will get the information about all the genes expression, but it's only one sample. If you want to look at the change of the gene expression, you have to sample different specimen. And however, in single cell analysis, within the one sample, you have thousands of cells, individual cells, and then use this particular cell barcoding technology. You can get information for thousands of cells, gene expression of thousands of genes. So this has become a matrix instead of vector of data points. So single cell gene expression analysis will give you the data, something like this. You have a lot of genes and you cross all the cells, and then its expression information is become available. Okay, so this become a big data problem because you have so many data points, and this gene expressed relatively high, and this gene expressed low, and some gene have very low level expression. But globally, if you look at the similarity between cells, so each point in this plot is a cell, and the cells forms different clusters. Okay, this could be a particular cell type, and all the cells are similar to each other, and all a group of cells have a similar expression profile across all the genes. They group together, so they're clustering problem, and they're also they're high dimensional, and a lot of nonlinear systems get involved. So we do need a machine learning to do this kind of analysis. So I have been working on this area for a couple of years, and we built a lot of tools around machine learning algorithm for single cell studies. And you can do this three-dimensional embedding and the pseudo time series analysis and build a network. We can also build a time series network. And there I did a lot of interaction tools, so you can use easy to use. And here, I don't think I have time to do the live demonstration, but if you look at the reference of this tool, you will get a lot of this. So you see here, I show you an example to visualize your cell and then start to drag and select, and then to know what the cell type is. So this particular example is from pancreatic cells. So there are alpha, beta, gamma cells in the sample, you can see here. Okay, so we developed a lot of machine learning tools, and there are some examples of this so-called single cell 24-net used to construct and compare single cell gene regulatory network. You can do virtual knockout, you can study the cell-to-cell communications, and look at their similar name because they are based on tensor decomposition and manifold alignment, so that's why I gave them a similar name. And this single cell GRN is for single cell gene regulatory network. And this is one so-called single cell 24-net. That's an overview about how this machine learning pipelines is built. So there are two samples of single cell RNA-seq data, and then do cell subsampling, and then construct network for each subsampled data. And then we do so-called tensor decomposition to denoise the tensor structure, and do this nonlinear manifold alignment to compare these two networks and to find the gene, so-called differentially expressed gene in the two networks. So we apply this method to some existing data set to demonstrate its usefulness. So this particular data is from a mouse brain, and this is a control sample, and this is a mouse after injection of the morphine, and see whether there's a neuron transcriptome difference. That's what we found using the tool I just described, so-called single cell 24-net analysis. Although differential expression cannot find anything, but we do find a gene set and also a network module which responds for the morphine addiction and the opium signaling. So that indicates our method is really accurate. We have a lot of such good examples to apply to different scenarios to use this machine learning tool to identify the different gene regulatory change between samples. So now we decide to use it to COVID-19 study. We download a set of data from the Nature Medicine paper published in 2020. So this data is called from a brochure alveolar immunocells from a COVID-19 patients. And they group those patients in two groups based on the severeness of their symptoms. One group is from mild to moderate, and another is severe group. and those people have less. It's only less than 20% of patients will develop such severe symptoms and in the critical conditions. But we're going to compare these two groups of patients and see what the gene regulatory difference between them, so we have a better understanding of the COVID-19 immune difference. So that's the data we get downloaded, and again, there's immune cells from patients, and then red cells from severe patients. We have over 3,000 cells, and from a mild symptom patients, they're the blue cells, they're 1,100. Using so-called marker genes, because there's some genes we know they're only expressed in macrophages or dendritic cells. We can confirm most of those cells are either macrophages or dendritic cells, they are immune cells. And then apply our tool, we can identify the highly significant genes. That's a table out of the comparison, that those change of the gene regulation is significant between mildly and severe condition patients. There are 27 of them. And then we use a technology called gene set enrichment analysis. Instead of only look at the most significant genes, we look at the whole rank of the list of genes. In this study suggests that there are 8 significant gene pathway. Signaling pathway is a significant difference between these two groups of patients. More interestingly, there are malaria and Pathogen, these two pathways were found to be significant in addition to this interfering and the signaling, and we know there are immune signaling pathways, so we expect to see those, but this too is give us surprise. So those are the genes, 14 genes and 71 genes in these two significant pathways. And why we think it is significant because nobody expected the difference between severe condition and the more mildly affected patients differ by malaria pathway. So that's the difference of the gene expression of those genes, green and red, which significantly differently expressed in two groups of patients, either upregulated or downregulated in a different direction. So that's a malaria pathway, and that's where the differential expressed gene located. If you only look at the differential expressed gene, then you only be able to find this pathway because they are not consistent pattern, not always upregulated or downregulated. It's a mixture of the different regulation directions. So that's why we need the network approach to identify this pathway. It's important because there are implications. People has already suggested the anti-malaria drug like hydroxychloroquine may be used to prevent and treat COVID-19. But this suggestion is under intensive debate. There are people agree and support this. A lot of other studies say, oh, no, there's no evidence. But in our analysis, we found this malaria pathway and the Pathogen pathway. That seems to suggest that maybe anti-malaria drug is useful, will be useful to control the more severe condition. And also this drug hydroxychloroquine, people know a lot of this mechanism because it has been used as not only anti-malaria drug for 60 years safely, but also as a drug to anti-rheumatology in rheumatology. So the mechanism at the cellular level we know is inhibit immune activity by reducing toll-like receptor signaling and the cytokine production. At the molecular level, it interferes with lysosome activity and autophagy. So in our study using machine learning tool, we found, okay, this malaria pathway and the VIBS pathway are maybe play a very, very important role in determining whether the SARS-CoV-2 infection disease can be developed into more severe condition. But in this case, if we have anti-malaria drug hydroxychloroquine applied to the early stage of the infection, maybe we will be able to control for the development of the disease. And we published this preliminary study in a preprint server. And if you are interested in, you can take a look. And that's all for my lecture. And thank you very much for your time.